Hello there. This video matches up with the spec point 3.7.1 inheritance, and particularly the section of sex linkage. So in the last video, we looked at a type of inheritance known as codominance, where you have two dominant alleles for one gene. And we also looked at a feature of it known as multiple alleles, where there are more than two alleles for the same gene. Now we're going to look at something known as sex linkage. So for us to understand this, we again need to go back to GCSE content, and we need to remind ourselves of sex chromosomes. So for a male, the chromosomes are X and Y, and for a female, the sex chromosomes are XX. Now, if we were to draw a Punnett square of the chromosomes for a male and female, we should come to the conclusion that for a male and a female cross, there is a 50% chance that the offspring will be female and a 50% chance that the offspring can be male, or a ratio of 1:1. And this is always the case. Now, the reason why we are looking at sex chromosomes is because, in fact, there are genes for specific characteristics that are present on the sex chromosomes. However, we must look at the structure of these chromosomes before we delve any further. So if we were to draw out both an X and a Y chromosome, we notice that the X chromosome is larger than the Y chromosome, and it's essentially cut in half. Now, the implications of that are if the genes that we are interested in, or this specific gene, is located on the X chromosome, because there's a half of a chromosome missing that's not on the Y. So that means that there is not a homologous portion, which, if it was a dominant allele, mask the expression of a recessive allele. So this can lead to some diseases. Now, the best example of this is hemophilia, which is a condition which leads to severe bleeding. Now, this condition is common in the royal families of Europe. So quite a number of members of the family have this condition or are carriers of this condition. So let's have a look, using our knowledge of Punnett squares, how this can get passed on. So if we take a scenario where we have a hemophilic male and we cross that with a normal female. Now, when we mean normal, we are saying a carrier. So normal being unaffected, however, they are carrying an allele for this disorder. So let's check our genotypes and see what they are. Now, because we are dealing with sex chromosomes, we have to differentiate between an allele for the disorder and a normal allele. So how we have to use superscript, and we did cover this in the lesson on codominance. So please have a look back at that video if you are non-sure about the superscript notation. So doing our normal sex chromosomes, XX for female and XY for male, but this time, because we are using a carrier female, we have to denote a capital H for normal allele and a lowercase h for hemophiliac allele. For the male, because they are XY, we only need to show one H. And because it's a hemophiliac male, it has to be lowercase h. So filling out our Punnett square for our offspring now, again, we're still using superscript. We have to do this for all of our offspring. We can see that we have two males, of course, and two females, of course. However, one of the females is a carrier and the other female is affected. For our males, it's exactly the same thing. But one of the males does not have hemophilia, but the other male does. The probability of having hemophilia is 50%, and the ratio is 1:1. Now let's take another scenario. This time we have a hemophiliac female and we're crossing that with a normal male. Using our Punnett squares again, we should now see a different ratio. Now we can see that in this scenario, our females are unaffected but carriers, but all the males are suffering from hemophilia. So our probability then of females having hemophilia is 0, because we have 0 unaffected, but they're all carriers. But for males, we have 100% of males that are having this disorder. And this is why a disorder such as hemophilia or most sex-linked disorders which are on the X chromosome are more common in males, because they do not have another X chromosome which can act as a mask for the recessive allele. So here is an exam question which I'd like you to try. So pause the video, have a try of the question, and then you'll see the answer afterwards. So for this exam question, we've got a pedigree diagram, which you may be familiar with in GCSE. So the key is given to us on the left-hand side. It tells us the situations for all parents and all offspring. So the first question is asking us to state the sex chromosomes of person B. So looking at the two genotypes for person A first off, we have an affected male, so that must mean they're first XY. However, because they're affected, it means that they have a lowercase h, whereas for a female, they are unaffected, so it needs to be XX, but they are not a carrier, meaning that both alleles are dominant, capital H. So to answer our question, why do the male offspring of A and B not have hemophilia, we can draw out a Punnett square to show this. So by doing a Punnett square, as we saw earlier, it has to be 2 males and 2 females. But the genotype of the females is showing us that they are both dominant alleles, meaning that they are unaffected from hemophilia. And our males are getting all of the X chromosomes from the female. And if both of the X chromosomes are for unaffected condition, that means all of the males will be unaffected also. And that is the reason why all of the males do not have hemophilia from A and B. And then for the last question, it says, using the Punnett square, calculate the probability that individuals C and D could have a child with hemophilia. So looking at the key, C is an unaffected male, which means that their genotype should be capital X, but the superscript should be capital H and Y. Whereas we have for D, we have an carrier female, which means that one copy of the X chromosome is affected and one copy of the X chromosome is unaffected, so heterozygous. Doing the Punnett square from that, then we should be able to see we get 4 different outcomes. We get 1 female which is completely unaffected. We get 1 female which is a carrier. We get 1 male which is unaffected. And we get 1 male which is hemophiliac. So that means that our probability of an offspring being hemophiliac is 1 out of 4, 25%, or 0.25.